So again, the human nose is extremely sensitive to sulfur and nitrogen-containing compounds. And because you can form a gas out of these thiols, they're added to natural gas so that you can smell them. Again, properties: foul odor. The scent of a skunk is due to thiol components. Again, sulfur makes things bad smelling, and people are sensitive to it at very, 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 very tiny amounts. So here we see the major components of the hog-nosed skunk. And notice, here we have our SH group. It's a thiol. And here we have a thioacetate. And so it's like acetate, but sulfur is in the place of oxygen. So again, thio, as you see here, THIO, means there's a sulfur in a place where you would have expected to find an oxygen. They have a lower boiling point than alcohols because sulfur does not hydrogen bond. Only nitrogen, oxygen, and fluorine does hydrogen bonds. And sulfur is readily oxidized to form disulfide bonds. And that's how we perm hair. You first put a reducing solution in someone's hair, and it breaks all the sulfur-sulfur disulfide bonds into SH groups. So you start with your disulfide groups, and then you put a reducing solution to get SHs back. You restyle a person's hair so the SHs are touching different SHs, and then reoxidize so those sulfurs lock together, and they're permanently locked together, which is why they call it a perm. And so a lot of proteins use this too. They form sulfur-sulfur cross bridges because sulfur is in a good range where it's easy to oxidize and reduce, so it's very easy to work for the body. So again, thiols form important biological molecules. The amino acid cysteine has an SH group, a thiol group, and that's really important for your proteins. The disulfide bond in insulin helps create the three-dimensional shape of the protein. And a thioester group is found in acetyl-CoA, which is an extremely important molecule if you study biology. Fats are synthesized out of acetyl-CoA. Glucose is broken down to pyruvate in glycolysis, and then it's oxidized to acetyl-CoA, and then acetyl-CoA enters a citric acid cycle. And so this molecule comes up a lot in biochemistry. So that thioacetate, or that thioester, is extremely important. So write the equation for the oxidation of T-butyl sulfide to T-butyl disulfide. Well, two of these link up, and both sulfurs bind to each other to form a disulfide bridge. And that's how they oxidize them. To reduce them, if you got a reducing agent, you could break them back into two thiols. And that would be reduction. And that is the end of this chapter.